Welcome to the video series on biotechnology and molecular biology. I'm Pranav Doradla, and today we're going to be talking about proteins, part 2. So today we're going to be talking about the second part of proteins. You can see that circled on the slide over there. Now, non-ribosomal peptides are smaller than proteins. They can contain weird amino acids that are not part of the normal 20 proteinogenic amino acids. They can be cyclic, which means that they're a circle, right? Instead of just being one chain, the chain is linked at its ends. And they can also be cross-linked, which means that there's covalent bonds between different parts of the protein, and that helps it hold its structure. So for many non-ribosomal peptides, they have inflexibility due to this, and this makes them good drug targets or good drug binders because they hold whatever conformation they've been designed for. And you can see examples of that on the side over there. Antibiotics, immunosuppressants, other stuff. They're synthesized by non-ribosomal peptide synthetases. So that's why we call them non-ribosomal peptides, because unlike normal peptides, they're not synthesized by ribosomes. Now, NRPS is a multi-enzyme complex, so it's many different enzymes placed together with different domains. The first step of synthesis for non-ribosomal peptides is called initiation. So A1 and A2 activation domains bind to and then activate the amino acid by adding a chemical tag to it. The editing domains then edit it, they add different chemical tags. The next step is elongation, where the C domain forms a peptide bond between one amino acid and the next. And then finally, with termination, the TE domain and other domains help release the formed peptide. So that's all for non-ribosomal peptides. Now we're going to talk about post-translational modifications. So after a protein has been translated by a ribosome, its job, it's not ready to go out and do its job in the world. It needs help from different modifications. So there's examples on the slide over there. I'll just go through each one. Hydroxylation is the attachment of an OH group to the side chain of a protein. This helps with solubility and different structure and functional elements. Methylation is adding a methyl group, which is CH3, usually at lysine and arginine residues. Residues are what we call the amino acids once they've been integrated into a peptide, because they're not really amino acids anymore. They're part of the peptide. They're the residues of amino acids. So methylation helps with protein-protein interactions and chromatin modification. Chromatin modification, if you remember, is a way to change how much a certain gene is transcribed. Lipidation attaches a lipid. This is also called prenylation, and it attaches a lipid. So lipids are like fats, basically, or fatty acids, and it attaches this to a protein chain. This helps with protein-protein and protein membrane interactions. The membrane of the cell, the outer casing of the cell, is hydrophobic, which means it's nonpolar, and fatty acids are also known to be very, very hydrophobic, very, very nonpolar on one end of them. So that allows the protein to stick into the membrane, and then if it has to do things within the membrane or outside of the membrane, but it has to be connected to the membrane, it can do those things. Acylation adds an acetyl group to the N-terminus, which is the amino terminus of a protein, or at lysine residues. And this helps with chromatin modification and also edits enzyme activity. Then disulfide bonds covalently link the S atoms, the sulfur atoms, of two different cysteine residues that we talked about before, that helps with folding. Sumoylation adds a small protein sumo, small ubiquitin-like modifier to the protein, and this helps with transport and transcriptional regulation, as well as a bunch of other things. Ubiquitination works with the ubiquitin proteasome pathway that we talked about earlier, and that obviously degrades proteins. Glycosylation attaches a sugar, so um, to an N or an O in an amino acid side chain, and this helps with stability, folding, cell-to-cell adhesion, as well as a bunch of other stuff. Phosphorylation adds a phosphate, so PO4, I think 3 minus, to certain residues on a protein, and this either activates or deactivates an enzyme by changing its energy. So the functions that I talked about here are definitely not exhaustive. These modifications do a lot of other things, and many of which are not even understood fully. But I know that was a very, very quick overview of these things. What I would suggest is you take your own time, go on Wikipedia, go on different websites. The internet is full of resources regarding this material, and spend some time just reading up on each of these. So with that, happy learning.